Okay. Today we will cover chapter 3. Chapter 3 is proteins, carbohydrates, and lipids. So usually if you go to, so I teach a protein engineering course for 1 semester. At that time, for 1 semester, I teach protein. But today, I will briefly explain about protein. In addition to protein, I will explain the carbohydrate and lipid. This chapter is composed of 4 subjects. First one is macromolecules. Macromolecule means they're large molecules. After that, we will cover protein, followed by carbohydrate. Finally, we will cover lipids. Okay? So first one is we will cover the macromolecules characterize living things. Okay? So macromolecule means that large molecule. Okay? So macromolecules function is dependent on the property of functional group. So this OH, we will call it as a hydroxyl. So this is the alcohol, the macromolecules, they have OH group. Okay? Their property is that they are polar and they forms hydrogen bonding with water, something like that. Okay? Okay, so this is we call it as aldehyde. Okay, aldehyde. Okay, and this the macromolecule, we call it as the acetyl aldehydes because they have the carbonyl group, the O group, so they are very reactive. So acetyl aldehydes, they are very reactive based on because they have the aldehyde group like this. Okay. And also we already know that carboxyl group is that like this. Okay, carboxyl group, this one. Okay, so this one is all of the acidic component has this carboxyl group. Okay, how about the amino group? R, N, H2, amino group. So all of the amino group is very basic, and all of the amino acid has the amino group. And also there is the phosphate and the SH group and also methyl group. Okay? So you don't have to memorize all of their function and property. Okay? Just cover. Okay? So each functional group has a specific property. For example, aldehyde group, they are very reactive, and the carboxyl group, they are very acidic, like this, okay? So next one is we will cover isomer. Okay, so during my protein engineering course, I teach this concept for 1 hour, but today I will briefly mention about the isomer. Isomer, in Korean we call it as 이성질체. 이성질체. 은세야.
예.
뭐 아무것도 없어? 예. 천재야. 책도 없고 뭐 아이패드도 없고 뭐 아무것도 없어?
책은 아직 안 들어가서.
책은 뭐 필요 없지 뭐. Okay. So isomer means that this is their terminology. Isomer is molecules with the same chemical formula, but their atoms are arranged differently. Okay. So first one is structural isomer. This is butan. How many carbon is there in butan? 1, 2, 3, 4. How many hydrogen they have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay? 4 carbon and 10 hydrogen. Okay, this is butan. How about the isobutan? They have the carbon is 1, 2, 3, 4. How about the hydrogen? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so they have the isobutan also has the 4 carbon and 10 hydrogen. Okay, they have the same chemical formula, but their atom is arranged differently. Okay, so there's two kinds of the isomer. First one is there is 3 kinds of the isomer. First one is a structural isomer. Okay, so structural isomer means that differ in the how atoms are joined. So their chemical composition is the same, formula is the same, but they are differ in how atoms is there. The joining method, the arrangement is different. Okay, so this is a structural isomer, and also there is a cis-trans isomer. So cis-trans isomer. Okay. The important thing in cis-trans isomer is that around the double bond, the carbon has double bond. Okay, so around the double bond, okay, this is also carbon also has double bond. So around the double bond, okay, in this case the cis-butene, okay, they have the in the day arm they have the methyl group and they have a methyl group. How about the trans-butene? So in the one arm they have the methyl group. One leg instead of the arm they have the one leg they have the methyl group. So this is a trans-butene. So cis-trans isomer means that different orientation around double bond. Okay, different orientation around the double bond. Okay, so this case is recorded as a cis, and this case is recorded as a trans. Okay, and optical isomer is the mirror image. Okay. Okay. So like this. So if you see, for example, if this finger is like this, the mirror image is like this. Okay. So this is amino acid. Okay. So their the mirror image is already different from the original image. So during the protein engineering course, I will teach is R isomer and S isomer. But today I will not cover in details, okay? Okay, the important thing is isomer. Korean we call it as 이성질체. There are 3 kinds of the isomer. First one is structural. Second one is cis-trans. The final one is optical isomer, mirror image. Okay, so during maybe when you are in 30 years, you will take the protein engineering course. At protein engineering course is also English class. And also at that time, I will teach you this concept for 1 hour. Okay, so today I will not cover in details. Okay. So now we finally cover in the first chapter, we will cover the condensation reaction and the hydrolysis reaction. Okay, condensation reaction, in Korean we call it as 축합 반응. hydrolysis reaction, in Korean we call it as a 가수 반응, okay, 가수분해 반응. Okay. There are two kinds of the reaction. One is the condensation, the other one is the hydrolysis reaction. Okay. Condensation means that two monomer, because the two monomer becomes polymer. Okay, that this polymer bind to another monomer, they will make polymer. It's like this. Hydrolysis means that starting from the polymer, after that it will make the monomer. That this one will also make the monomer. This monomer is like this. Okay. So condensation means that they combine to each other. So monomer and monomer. So during monomer and between monomer, energy is used to make the covalent bond to make the polymer is like this. During that time, always the H2O is made is like this. Okay? H2O is made. After that, this polymer and this monomer, they combine to each other. At this time is also another covalent bond is made, and H2O is produced is like this. Okay? Hydrolysis reaction means that this polymer is broken down into monomer or intermediate, and this intermediate is broken. But at that time, usually the H2O, they used H2O, so this covalent bond become the OH, and this one become the H is like this. Once again, this covalent bond, they will consume the H2O, and this one becomes OH, and this one will become the H, it's like this. So we have the two reactions. First one is condensation. So during the condensation reaction, macromolecule is made is like this. And during hydrolysis reaction, macromolecule becomes the monomer is like this. Did you understand it? Okay. Now we will cover second chapter, protein. Okay. So if you are interested in protein engineering or protein, okay, so I will teach you this subject for one semester, okay? Okay. Okay. So let me start. The basic subunit of the protein is recorded as amino acid. So amino acid is monomers that make up the protein. They have a 4 functional group. Starting from the amino group and carboxyl group and hydrogen and side chain. Okay? So based on their location, so we can make the R configuration and S configuration. The, amino acid, they show that the optical isomer, but today I will not cover in details. Okay, but they have the 4 functional groups, starting from the amino, carboxyl, and hydrogen. Finally, they have the R side chain, it's like this. Okay, and they have 20 amino acid. Okay, starting from the alanine, and there are lots of the amino acid. So during my protein engineering course, I always said to the student, memorize all of structure. Okay? And they also have the simplistic code. For example, arginine, we call it as R. And histidine, we call it as H. Lysine, we call it as K. So lysine they have this kind of R group. They have different R group is like this. So if you are interested in the amino acid, you had better take another course, okay? But today I will not cover in details, okay? But the important thing is that, okay, this amino acid, they have arginine and histidine and the lysine, they have the positive charges. Okay, how about the aspartate, aspartic acid and the glutamic acid? They have a carboxyl minus group. Okay, and they also have the final one is they have the R group with the hydrophobic side chain starting from the alanine, leucine, isoleucine, leucine, something like that. Okay. So if you see lots of the alanine, isoleucine, and leucine, something like that, this is the R side chain is that they're hydrophobic. So if you find that there are lots of the alanine, isoleucine, and leucine, it means that this protein is located based on this information. Maybe this amino acid is located in this region. Okay. If they have the arginine and or histidine or the lysine, you will find this kind of things here. Okay. But today I will not cover in details. Okay. And also they have the R group with hydrophilic, and also they have a special case injuries like this. Okay. Okay. Okay, you don't have to memorize. Okay. Okay. Okay, the simplest amino acid is glycine. They only have the H glycine. So whenever they have, because whenever there is, if you find the glycine, it means that between the glycine, because glycine is the very smallest one, so that location is rotated is very well. So we can predict it is like this. But I will not cover details. But also, if they have methyl group, this is alanine. Okay. Okay, so you don't have to memorize it. Okay. Okay, so in the amino acid, peptide bond formation happens. Condensation reaction is happen to make the macromolecules. Okay. So this peptide bond formation is condensation reaction, because they will produce the H2O. Okay, they will make the covalent bond is like this. So this bond we call it as a peptide bond. So peptide bond formation is formed by condensation reaction between the carboxyl group and the amino group. After that, water is produced, and they will make the peptide bond is like this. Okay. So maybe 2 years ago, during the midterm exam, I showed the one amino acid, and I will show another amino acid, and write down to make the peptide bond formation. So in the blank, you have to write the H2O, and you have to write down the peptide bond is like this. Okay. Okay. So peptide bond formation is also condensation reaction. Okay, it's like this. Okay, and also we will cover primary, secondary, tertiary, and quaternary structure. So during the protein engineering course, primary and second, during 1 hour I teach is primary and secondary structure. Because lots of information I have to teach it. But today I will briefly cover the primary structure of protein and secondary structure of protein. Okay, primary structure is the polypeptide chain. Okay, so they have the peptide bond here, peptide bond here, peptide bond here, and peptide bond here. Okay, so primary structure is polypeptide chain. Okay, stabilized by peptide bond, peptide bond, peptide bond, peptide bond, peptide bond, peptide bond is like this. Between the CO and H, there is a peptide bond. CO and H, there is peptide bond is like this. Okay. Secondary structure is, we already know that secondary structure is composed of alpha helix. Another one is a beta strand. Okay. So each one is that stabilized by the hydrogen bonding between the, this is the amino group and this is the carbon, oxygen. So between here, there is the hydrogen bonding is there. So usually the alpha helix is right-handed. So alpha helix is right-handed, it's not left-handed. So alpha helix is right-handed. So between maybe here there will be CO and the NH. Between them hydrogen bonding, hydrogen bonding, hydrogen bonding, hydrogen bonding is like this. If there is no hydrogen bonding, they will not go forward. Okay. And also this is a beta sheet. So they are also combined to each other by hydrogen bonding. Okay. Okay. But during protein engineering course, so each turn is composed of 3.6 amino acid, and we have to calculate the distance. But during the biology course, you don't have to. But we have to know that alpha helix and beta strand is stabilized by hydrogen bond. Okay. So in the homework, there is a question asking about hydrogen bonding. Okay, you have to know the hydrogen bonding. Okay. Tertiary structure. Okay. Tertiary structure means that they will forming specific shape is like this, composed of the alpha helix and the beta strand, but they will make the shape is like that. They will make this kind of shape, and they will make this kind of shape. The shape is like this. Okay. Okay. The most important thing in the tertiary group, usually between the SH, usually the disulfide bond is formed, is like this. Okay, between the SH, disulfide bond is happening, is like this. Okay, disulfide bond is forming, is like this. Okay. Okay. So to form specific shape, usually the disulfide bond is stabilized tertiary structure is like this. Okay. Okay. Okay, so our hair is also protein. We call it as a collagen. Okay. And some of our people hair, most of people hair is straight, but some of them they are curled hair. So maybe that person has lots of the disulfide bond forced to make the curl. Then there will be the disulfide bond. Okay, it's like this. Okay. And also, most important thing, disulfide bond, and also hydrogen bonding, and also between the hydrophobic region, there is a hydrophobic interaction which happens. Okay, after that, this is tertiary structure. Okay, so during the protein engineering course, we always calculate the power of the disulfide bond and the power of the hydrogen bonding and power of hydrophobic interaction. But today I will not teach you the specific information. Okay? Okay, the important thing is tertiary structure, especially stabilized by the disulfide bond. Okay, it's like this. Okay, the final structure is a quaternary structure. Okay, this is the hemoglobin structure. They have 2 alpha and they have 2 beta. Okay. So quaternary structure means that 2 or more polypeptides assemble. 2 or more polypeptide means that 2 or more tertiary structure assemble. This is tertiary structure, and this is tertiary structure. This is tertiary. They assemble to form large protein molecules. Okay, this one we call it as hemoglobin. So this is hemoglobin structure like this. Okay. Okay, at that time, stabilize that hydrogen bonding, disulfide bond, and hydrophobic interaction, and ionic bond is also involved in this interaction. Okay. Okay, quaternary structure. Okay. Okay, so if you see details. Okay. Okay. Disulfide bond is covalent bonding, 공유결합. However, the hydrogen bonding is hydrophobic, and ionic bond we call it as non-covalent bonding. Okay. For example, between the carboxyl group and amino group, there is an ionic bond is happen between the charges R group, but this is not covalent bonding. This is non-covalent bonding. They do not share the electron. Okay. So hydrophobic interaction means that occurs between 2 non-polar groups. Okay. And also hydrogen bonding between the hydrogen and also another group is like this. Hydrogen bonding occurs between the negative charges and between the positive charges on the hydrogen atom is like this. Between them, the hydrogen bonding is happening. But this one is non-covalent interaction. However, disulfide bond they share the electron together. So we call it, in Korean, we call it as 공유결합. Okay. Okay. Stabilize is like this, okay? Okay, and this is the figure 3.11 to show that primary structure specifies tertiary structure. Okay? So this is the tertiary structure of proteins. Okay, they are composed of the alpha helix and the beta strand. It's like this. So we can extract and we can only purify this protein. Okay, so this is especially the ribonuclease. Okay. So because I teach is protein engineering course, so I know whenever I see the structure, I know that this is nuclease because they have their shape. They have this shape. This shape is they can bind to DNA. And they can digest it. The DNA is like what today I will not cover. So this is the functional tertiary structure. After that, we can add chemicals to disrupt hydrogen bonding, also disrupt ionic bonding, or disrupt the disulfide bonding. After that, slowly remove the chemical reagent. So when the disruptive agents are removed, this three-dimensional structure is restored. Okay. Okay. For example, after we did perma, so our hair becomes curled, but several times later, 1 month later, our hair becomes straight, is like this. Okay. Okay. So primary structure. So each primary structure, for example, between here, the primary structure they have the cysteine, they have the SH group, and this primary structure they have another cysteine group. So this primary structure between them, they will make the disulfide bond. After that, they will make this kind of shape is like this. Okay. So this experiment to show that primary structure specify tertiary structure, or primary structure can specify the quaternary structure. Okay. Okay. And also protein shape can changes as a result of interaction with other molecules. Okay. For example, so one enzyme is the bind to another protein. After that, usually the enzyme shape is changing. To digest this protein, usually the enzyme shape is changes like this. Okay. So binding protein shape is changes as a result interaction with other molecules, or okay, this amino acid. Okay, because you don't know the structure of the protein, but if they have the lysine, we call it as K, or so in specific location of amino acid, for example, phosphate can be added to this kind of amino acid. So after that this protein molecule can be changed like this. Only one phosphate is added, so they can be activated. 좋은 소리 알겠어? Okay. It can be activated just like this. Okay. Okay. So the proteins. Okay. So okay. And also environmental condition affect protein structure. The most important thing is the temperature. Okay. Usually the egg is composed of, most of them is composed of the protein. Okay. So after we increase the heat, increase temperature, the protein can be denatured like this. Or another environmental condition is changes or chemical reagent can change their protein structure. So this is one example. Okay. Okay, so protein can be denatured, but denatured proteins can be changed to their original structure. Chaperone proteins. Okay, chaperone proteins. 아상현 관리하고 있어? Chaperone. In Korean, chaperone. What is called chaperone? 보이죠? chaperone이 무슨 말로 보이죠? 보호자. chaperone is we call it as 보호자. So chaperone, after chaperone found the denatured protein. Okay. So 보호자 found denatured protein. After that, so they have a lead. They have the lead pack. After that, this chaperone protein will pulse and make the original form is like this. Chaperone is also like the teacher. Okay. So during the lecture, whenever until fall asleep, I wake up. Okay. 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 So usually I teach proteins for one semester, but I briefly summarize the proteins. Okay, starting from the amino acid, the four functional group, and also their peptide bond formation, and primary, secondary. But you have to know the important hydrogen bond, bonding. Okay, tertiary and quaternary. Okay, and also based on this experiment, we can say that primary structure specified tertiary structure. So in Korean words, similar one, 습관이 운명을 좌우한다 있죠, 맞죠? 그렇게 산 거예요. In Korean, okay. And also protein can changes. Also, environmental condition affect protein structure, but chaperone protein found and bind to the denatured protein and help them to refold into the original shape. Is like this. Okay. Okay. Let me go to the third subject, carbohydrate. Okay. In Korean, we call it as 탄수화물. Leah and Marie, did you understand it? Okay, my lecture, okay? Whenever you have a question, please let me know. Okay? So their main structure is like that. like this. So if they have the 5 carbon, so they will be like this. If they have the 6 carbon, they will be like this. Okay. Okay. For example, this is the glucose. Glucose has the 6 carbon. 1, 2, 3, 4, 5, 6. Okay. Based on this formula, we can assume that oxygen, we can find oxygen is 6. 1, 2, 3, 4, 5, 6. How about hydrogen? So we can find 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 is like this. So carbohydrate is they have their structure is they can have the straight form, and also they can have the intermediate form without bonding here. So they did not make the ring, but they also can make the the ring is like this. Okay, ring structure is like this. But they are composed of the alpha orientation and beta orientation. Okay, so based on this carbon is first carbon, first, second, third, fourth, fifth, sixth carbon. Based on the first carbon, if the OH group is go downward, so we call it as alpha orientation. If this OH group is go to the arm, so we call it as beta orientation. Okay. So when dissolved the water, the alpha and beta form of glucose, they interconvert. They can be interconvert. Okay. Okay. Carbohydrate is the source of the energies, and their main structure is like this. Main chemical formulation is like this. Okay, they can have the straight form and intermediate. They have alpha, they have beta form is like this. So we call it as alpha D glucose, and in this orientation we call it as a beta D glucose is like this. Okay, so now we will cover monosaccharide. And next slide we will cover disaccharide. Finally, we will cover polysaccharide. Okay, this is done. Okay, let me start from the monosaccharide. Monosaccharide means 1. Okay, 1. So usually if they have the 5 carbon, we call it as a 5 carbon sugar. For example, ribose. So we call it as a pentose. And also there is another example, 6 carbon sugar. They have 1, 2, 3, 4, 5, 6. So we call it as a hexose. Okay. In pentose, we can see that ribose. And if there is no oxygen, we call it as a deoxyribose. So, where can we find the ribose? RNA. RNA. Ribose is the main component of RNA. Where can you find the deoxyribose? DNA. Okay. And also 6 carbon. This is the alpha mannose, and this is the alpha. OH is like this, alpha glucose, and this is the fructose is like this. Okay. For today's is just a biology class, so you don't have to memorize structure. Okay. Okay. This is monosaccharides, but the important thing is 5 carbon and 6 carbon. And disaccharide, okay, to make the disaccharides, usually the glycosidic bond is happen. So glycosidic bond is a condensation reaction or hydrolysis reaction. 터지는 거야. glycosidic bond, to make the glycosidic bond, so to make them, the condensation reaction should be happen, or hydrolysis reaction should be happen. Condensation. Okay, to make the one molecule become two molecules like this. One molecule become the polymer macromolecules. So this is also glycosidic bond is also condensation reaction. So between the two monomer, so glycosidic bond is happens. So they produce H2O. So they will make the sucrose is like this. Okay, so this is alpha D glucosol. OH is like this, and this is fructose. So between the alpha, this is the first carbon, and this is second carbon. Between 1 and 2, alpha 1, 2 glycosidic bond is happens like this. Okay, and also alpha D glucose and beta D glucose, they combine to each other. After they will make the maltose. Okay, this carbon is first carbon, and this carbon is fourth carbon, and this OH is the alpha orientation. So between them, alpha 1,4 glycosidic bond is happen is like this. So this is recorded as a maltose. So another also very famous example is the cellulose. So we call it cellulose. So beta D glucose and beta D glucose, they combine to each other. At that, they will make the beta 1,4 glycosidic bond is like this. So they will make the disaccharide. Okay. So glycosidic bonds between two monosaccharides can create many disaccharides. Okay. So this is the in the midterm exam. So I can show. I can ask you one of question. Okay. Okay, so this is the beta 1,4 glycosidic bond. You don't have to memorize the structure, but you have to know the location. Alpha, beta is like this, okay? And poly means that large polymer of monosaccharides are connected by glycosidic bond. Okay, the first one is, so large polymer means that large macromolecules. The very famous example is cellulose. So they are composed of the beta 1, alpha, beta D glucose, beta D glucose, beta D glucose, beta D glucose, beta D glucose. So between them there is beta 1,4 glycosidic bond. How about the glycogen and starch? Okay. So they are composed of the alpha 1,4, but some of them they have the alpha 1,6, the alpha 1,6 glycosidic bond. They have branches like this. Okay, this is the starch and also the glycogen. Okay, so
네.
what is cellulose?
셀룰로스.
Whenever we eat the parsley, all parsley is composed of the cellulose, and also another one is the another vegetable is composed. All of them is composed of the cellulose. Okay, so this is their structure is like this. 프로그램 잘 이해하고 있어? 김지민? Okay. But the cellulose, they don't have the branch structure. But this starchy and glycogen, they have branch structure. Okay. Okay, you will see here. So this is the cellulose. They don't have the branches. And this is starchy. But they have branches like this. And this is starchy. But glycogen is they have the higher branches like this. Okay. So this is starchy and this is glycogen. Their structure is the same, but the glycogen has more branches like this. Okay. Okay. The important thing is that whenever they make the disaccharides, glycosidic bond is happen. Okay. You don't have to memorize the structure, but you have to know alpha 1D and alpha 1 4 and beta 1 4 glycosidic bond. Okay. Okay. So let me go to the final subject, lipid. Okay. The basic function of lipid, they can store energy, and also they structural role in the cell membrane. Most of cell membrane is composed of lipid. Okay, so this is the basic structure of lipid. We call it as a triglyceride. Okay, they are composed of one molecule of glycerol. They have OH, glycerol. After that, this is the fatty acid. So finally they have carboxyl group, and carboxyl group, and carboxyl group. After that, they will make the triglyceride like this. This one is recorded as ester linkage. Okay, ester linkage happen. Between them, they will make the ester linkage. Okay, so they will make the H2O. It means that this reaction is also condensation reaction. Okay. And 1 year ago, I showed that in the midterm exam. So I give this structure, glycerol structure, and also give this fatty acid structure, and just write down the glycosidic, write down the ester linkage. So ester linkage is happen between this oxygen and this carbon, so they will make the ester linkage, acid linkage like this. Okay, the important thing is that they have the body, and they have three legs. It's like this. Okay, 1, 2, 3. Okay, but in the lipid, 1, 2, 3 can be leg, but sometimes okay. I will show you the final one. Okay. Okay, so this is and also this is fatty acid. They have the legs. Okay, this kind of leg. But there are two kinds of fatty acid. First one is saturated, and the other one is unsaturated. Okay. For example, the saturated, there is no double bonds, so they are very straight, like this. But whenever there is a double bond happen, so they will make the cork is like this. So unsaturated fatty acid is a 1 or 2 double bond in the carbon change. So they will make the cork is like this. Cork. We call it cork. Cork. So it can prevent packing. In this case, this is in in this location. 1, 2, 3, 4 can be located here. But this one is they make cork, so in this location, in this dimension, there can be only 1 and 2. But this one they can be packed, but it cannot be packed. Okay. Okay. So this is the example of saturated lipid, our the red meat, and also cheese, and also unsaturated fatty acid means that there is olive oil, and another more there is a more saturated unsaturated omega-3 is unsaturated fatty acid. Okay. Okay. So usually the unsaturated fatty acid, they are liquid at room temperature. Okay, but saturated, they are packing. So usually they are the solid at room temperature is like this. Okay. The difference is the double bonds. Okay. So they have 3 tails, but in these tails they don't have double bond, but in this tail they have the double bonds. Okay. That's why they will make this one is prevent packing. Okay. Okay, so let me start explain the phospholipids. Okay, so this is glycerol. Usually lipid, they have 3 rags, fatty acid. But sometimes these 3 rags become 2 rags, and one half is like this. Okay. So usually phospholipids, 2 fatty acid is bind to the glycerol. So this linkage, we call it as ester linkage. Okay? And after that, instead of fatty acid, phosphate group can bind to the glycerol. Okay? The phosphate group can replace 1 fatty acid. So this will become the head, and this one become the tail is like this. Okay? Because phosphate has minus charges, so usually this phosphate group is used hydrophilic because sometimes they have minus charges and sometimes they have positive charges. So the head is the hydrophilic. However, 2 tails, there is no charges. Okay, it means that they are hydrophobic. So that's why it can make the bilayer is like this. So this is 1 layer, and this is the other layer. Okay, head is they meet the water. Hydrophobic region is they combine to each other. There will be lots of hydrophobic interaction will be happening. Okay. Okay. Most important thing, head is hydrophilic and the tail is hydrophobic. Okay. Okay, so because of this structure, so they have the amphiphilic. So one is the hydrophilic, and one is hydrophobic. So they have the amphiphilic. So they like the water, and also they don't like the water. Amphiphilic. Okay, they have 2 kinds of the characteristics. Okay. So in water, usually tails stay away from the water, and head is interact with water. So finally, they will make the forming phospholipids bilayer. Phospholipids bilayer like this. Okay. Okay. So today we cover